Hello friends. In our last topic we studied about genetics, Mendelian genetics, and this is iBiocare classes that deals with NEET AIMS NISA Olympiad given by the teacher AKB sir, and you can visit the site to get details information, that is www.ibiocare.com. Let's start with the topic, that is how to find the genotypic ratio of a dihybrid cross without using the Punnett square. In the last topic we have discussed about how to find the monohybrid ratio, how to find the dihybrid ratio without using the Punnett square, and now we are going to find the genotypic ratio of a dihybrid cross without using the Punnett square. So we have already, in the last class, we have already studied about the genotypic, how to find the genotypic ratio of a monohybrid. I'm just revising it, just brushing it up. That is, this is the monohybrid ratio 1. In the dihybrid ratio, we find two character. So here what happened? This is the character of the tall. One is that is the we have taken the height of the plant here, which is of genotype ratio 1 is to 2 is to 1. This is 2 by 4. We have simplified this. This is 2 is to 4. We have simplified it into 1 into 2. Then this is the other character, that is the purple character. This is the color of the plant. This is the height. So we have taken two character. So as we have taken two character, so it is regarded as dihybrid cross. And the phenotypic character of this monohybrid cross are written as like capital T stands for tall, small t stands for dwarf, capital P stands for purple, and small p stands for white. Now let us move how to find the genotypic ratio by using the genotypic ratio of a monohybrid cross. So now we need to find the genotypic ratio of this particular dihybrid cross. Let's start. So without using the Punnett square, how to find the genotypic ratio? First we should write the genotypic ratio of the first character, that is height in the dihybrid cross. The first character is the height. So we should write the height genotypic ratio, that is 1 is to 2 is to 1. Then we write to the color genotypic character, that is purple. It is also what? Character is 1 is to 2 is to 1. So accordingly we have found out that tall, heterozygous tall, homozygous dwarf, homozygous purple, heterozygous purple, homozygous. Now what we do, first thing is that we should take one of them as constant. Suppose I'm taking this particular character as constant. So now I'll cross this with this, this with this, this with this. Then I'll cross this with this, this with this, this with this. Then I'll cross this with this, this with this, and this with this. Simply without using genotype Punnett square, I'll get the ratio. Let's start. This I'll cross purple, I'll cross with tall. So I'll get what? Now I'll get the ratio in the form as 1. This multiplied to this, I'll get 1. Got it? This multiplied to this, I will get 2. So this multiplied to this, I will get a 2. This multiplied to this, I'll get 1. Then this one multiplied to this tall, I'll get 2. This multiplied to this 2 plus 2, 2 into 2, I'll get 4. And this multiplied to this dwarf, this, I'll get 1. That is 2, sorry. So likewise, when I'll multiply this with this tall, this heterozygous tall and dwarf, I'll get this as the ratio. So accordingly, I got the ratios 1 is to 2. I got 1 is to 2 is to 1. That is first when I cross this one with this one with this one. Then I got 2 is to 4 is to 2. That is when I cross this with this with this, and this with this. Then I got 1 is to 2 is to 1. That is the last one. 1 is to 2 is to 1. So these are the genotype ratio accordingly. And I got the character also. I got this is pure. Here I got tall purple. And likewise I got one tall purple, I got another one, I got dwarf and purple. So easily without using the Punnett square, you can easily get the ratio with its character. So the genotypic ratio of a dihybrid cross is 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. So this is one of the simplest way to get the genotypic ratio while you are doing the problems in your entrance exam. This will save your time. Okay. Next, another easy method how to find it. That is, let's move how to find the genotype ratio dihybrid cross. Likewise, we know the genotype ratio of a monohybrid cross is 1 is to 2 is to 1. So first we'll write 1 is to 2 is to 1. Again we'll write dihybrid means 2 monohybrid. So again I'll write 1 is to 2 is to 1 on both the end. So in this end I'll write 1 is to 1 and this end I'll write 1. So both of them are the monohybrid ratio. One monohybrid will give the dihybrid. Simply what we do, we'll add this to this, add this to this, addition, add this to this, no multiplication. So what I'll get, 1 plus 1 I'll get 2, 2 plus 2 I will get 4, 1 plus 1 I'll get 2. So here also I got the genotype ratio of a dihybrid without using the Punnett square, and it is very easy way that will help in your entrance exam. Next friend, how to find the trihybrid ratio using the genotypic ratio, using the monohybrid ratio or a dihybrid ratio without drawing the Punnett square. How easily we can get it? So let's move. How easily we'll get it? First, we know friend that the dihybrid ratio is what? 9 is to 3 is to 3 is to 1. It is the phenotypic ratio. Then friend, we know the monohybrid ratio of the phenotype ratio is 3 is to 1. Very simple. Here what we have to do, simply multiply 9, 3, 27. 3, 3 into 3, 9. 3 into 3, 9. 3 into 1, 1. 1 into 9, 9. 1 into 3, 3. 1 into 3, 3. 1 into 1, 1. Simply we'll just multiply it and we'll get the trihybrid ratio accordingly without going into the Punnett square. So first 27, 9 into 3, 27, then 9, 3 into 3, 9, then 3 into 3, 9, then 3 into 1, I have accordingly sorted out, got it. So I get 3. This is the trihybrid ratio. Simply using the dihybrid phenotypic ratio and monohybrid phenotypic ratio without using the Punnett square. This is how easily you can solve your problem while going into entrance exam. Next friend, we should know what is backcross and what is test cross. So first we'll know what we do in a backcross. In backcross, what happen? Normally in a backcross, what happen? Now we always cross. This is the parent with the any of the parent with F1 generation. Either you can take this as the parent or recessive as the parent, and you cross when you cross them with this F1 generation, you will get the backcross. That is, suppose I have taken the parent, one of the parent, this is the parental character, capital T, or you can take small t accordingly. And this one is the F1 generation character, that is capital T small t. In the F1, when we cross this both two, we'll get big T small t as the in the F1 generation. So when we cross them, we got homozygous tall, homozygous tall, heterozygous tall, heterozygous tall. So all 100% tallness will get. So in backcross, what happen? We'll always get the 100% tall. So we get 100% tall of both homozygous and heterozygous, each with 50% character with ratio as 4 is to 0. Normally backcross are used only to get the backcross is to know the phenotypic character. Let's talk about the test cross. Now, what do you mean by test cross? In test cross, what happen? Normally, what happen in test cross? We always cross the F1 generation. F1 generation, we always cross F1 generation with the recessive parent. With the recessive parent. We always cross with the recessive parent. This is very important. Recessive parent. F1 cross with the recessive parent. We know these are the recessive parent, small t, and these are the F1 generation. So this is the F1 generation, friend, and this is the parental character. So when we cross, we got what? Now we got heterozygous tall. This is heterozygous tall, that is 1 and 2. And we also got homozygous dwarf, that is also true. So in the ratio we get 2 is to what? 2, which equals to 1 is to 1. we simplify, we'll get the ratio accordingly. But here we get the ratio in back cross, we get the ratio as 4:0. So in a test cross, what happened? We get 50% tall and 50% tall, so that the ratio is 2:2, that is 1:1. Always the test cross implies 1:1 ratio. And it's very important to know that the test cross is used to determine whether the individuals exhibiting dominant character are homozygous or heterozygous. So normally test cross are used in your problems to know the dominant character of a homozygous or a heterozygous form. So this is all about backcross and test cross. In backcross we'll get 4:0 is the ratio, whereas in test cross we'll get 1:1 as its ratio. So friends, this is what we know, and in our next video we'll learn about Mendelian law with some problems. Hope you enjoy it, and in a further video I'll give you problems and I'll make you understand the tricky way, easy way to know the problems. First we should know the theory, and knowing the theory details, it will easy on part of us to know the problems or to do the problems easily in the entrance exam. So friend, you can visit our site and you can get all information there about your entrance exam questions online class. Okay, thank you.